Okay, hello and welcome to Shrawan, Alex Kuznaratis and Jacob's process of transcription video. First of all, you start with the DNA. If we have a look closer, the DNA unzips itself into two strands of straight DNA, bonded with hydrogen bonds. As we come along, mRNA begins to form and the hydrogen bonds are broken. And this is what we get as a result. The mRNA begins to create complementary bases for each one of these bases. It copies the DNA of these to form something similar to this. Now the only difference is C bonds with G, G bonds with C, and A bonds with U. Thymine has been replaced with uracil. Now, after that, the mRNA is formed along here and leaves in a gap in the nucleus and heads towards the ribosome. So this leaves out of the nucleus and heads towards an organelle in the cell. The DNA rezips itself to form the complementary bonds, and the complementary bases and the hydrogen bonds are replaced in between. It then re-swivels itself back to DNA. Thank you very much for watching. It's been
Transcription takes place in the nucleus of the cell. The weak hydrogen bonds in the DNA are split and one strand is copied. The copy has replaced thymine with uracil. This copy, or mRNA molecule, leaves the nucleus and attaches to a ribosome. Part 2. Translation. A codon is a group of 3 bases that code for a specific amino acid. A tRNA arrives at the ribosome. It holds a code complementary to a triplet code of the mRNA and the amino acid it codes for. The tRNA breaks away and leaves the amino acids joined together.
Translation is the process of the formation of a specific protein coded by the DNA. The process starts with the mRNA made in transcription. The mRNA moves out of the nucleus in a cell through the nuclear pore into the ribosome located in the cytoplasm. The mRNA enters the ribosome where the bases are read in sets of 3, known as codons. As the ribosome reads the codons, it creates a tRNA molecule with complementary base pairs to the codon. The tRNA molecule takes the bases on the codon and codes them into a specific amino acid decided by the order of the bases. As the tRNA for the next codon arrives, the amino acids form a bond and the first amino acid attaches to the second and the tRNA for the first acid leaves. This process continues and the amino acids continue to chain together. This forms one chains of amino acids also known as polypeptides which make up proteins. And this is how specific proteins are formed from code from the DNA. A chain of over 50 amino acids is formed.
Protein manufacturing happens in body cells. Here is our nucleus, and here is the cell membrane. What's inside the nucleus? In the nucleus, there are 23 pairs of chromosomes. These are represented as socks.
First, we need our chromosome.
Inside the chromosome is a string of DNA. This brings us to stage 1. Transcription.
Here's a string of DNA. Let's take a closer look.
DNA is structured in a double helix.
The double helix structure is connected together by weak hydrogen bonds.
The two strands are made up of sugars and phosphates.
The strands are connected by complementary base pairs.
Thymine,
Adenine.
Cytosine and guanine.
Here is our enzyme, RNA polymerase.
He is used to break the hydrogen bonds, which separates the strands.
a single strand is produced by reading one side of the separated DNA and matching it up with its complementary base pairs.
This strand is called mRNA, messenger RNA.
Why is this strand different to this strand? Uracil replaces thymine as adenine's complementary base pair.
This all happens in the nucleus.
The mRNA strand exits the nucleus via the nuclear wall into the cytoplasm.
Welcome to the cytoplasm.
In the cytoplasm, the mRNA strand attaches to a ribosome.
We have now finished stage 1 of protein synthesis. This leads us on to stage 2, translation.